Hello students, today we are going to discuss the chapter seed. We have already finished with pollination and fertilization. So today we are going to discuss about the product of fertilization, that is seed. We all know that after fertilization, post-fertilizational changes takes place. In this post-fertilizational changes, the seed is produced from the ripened ovule, whereas fruit is produced from ripened ovary. So if you remember this carpel-like structure, in the carpel we are having a stigma, style, and ovary. Inside ovary an structure is present and ovule is present actually, which is then converted to seed, and inside this the zygote is developed. That zygote will be soon developed into an embryo. So ultimately embryo formation is taking place inside this ovule. Then what is the structure of embryo? It consists of which parts that we are going to discuss today. If you remember that sepal, petal, androecium means stamen, and this is stigma and style. This part, this part, this part, this part, and this part will dry and withered off, means fall off, and only this ovary part of the flower will be converted to a fruit. If you want to convert it to a fruit, imagine it like this. And in case of brinjal, if you remember, we are having persistent calyx. So in that case, you will be having this kind of a structure which is converted to brinjal. The ovary part is this, and this is calyx which is persistent in case of brinjal. Let's move to the diagram to understand it in a better way. So if you are proceeding with this structure, this is a small structure of ovule which is having micropyle, the opening. Then it will be having two integuments, means the covering which is making the seed coat respectively, outer testa, inner tegmen, and the tissues of the ovule are called as nucellus, and inside nucellus something is present which is called as seven-celled, eight-nucleated embryo sac. I already discussed structure of this in pollination chapter. So let's move with the types of seed and parts of seed. When we talk about parts of seed, each seed consists of two structures. One is called as embryonic axis and second is called as the cotyledons. So if you can see this seed, it is having the embryonic axis, and this embryonic axis is consisting of plumule which will form future shoot, and this radicle which will form future root. Now these two thick fleshy structure which you can see, these two structures are called as cotyledons or embryonic leaves, which will be providing nutrition to the embryo, and it will be, you can say, stored for even germination of the seed. When you talk about the types of seed on the basis of endosperm, it is called as non-endospermic and endospermic seed. We will discuss that later on. First, let's move with the structure of the seed. So this we have seen. The definition of the seed is the ripened ovule. The formation of seed after fertilization. So ovule after fertilization becomes seed, that we have done. And after fertilization, the ovary is forming the fruit, that we all know. Parts of seed, as I told, there are two parts: the main proper embryo which is having embryonic axis. So if I am drawing a rough diagram for you, let's see. This is the plumule and the lower portion is radicle. This thick solen structure which I am making, these are called as cotyledons. So the upper portion is called as plumule. This lower portion is called as radicle, and these two solen structure which is storing food is called as two cotyledons. Now these cotyledons are two in number, so it is called as a dicot seeds. The plant which consists of dicot seeds are dicot plants. Like all the pulses, you can write pea, gram, bean. And if it consists of a single cotyledon, as in case of maize, which we will discuss later on, is called as monocot seed. Now here we have to discuss the structure, like what is embryonic axis, what is epicotyle, what is hypocotyle, that we will see. So this portion, the portion of the embryonal axis, this is the embryonal axis which I am drawing, starting from radicle till plumule. This complete structure, the straight line, is called as embryonal axis. Now the portion of the embryonal axis which is above cotyledon, this portion is called as epicotyle, whereas excuse me, whereas the portion of the embryonal axis which lies below the cotyledon, this is called as hypocotyle. This is called as hypocotyle. So these five terms we should always remember whenever we are talking about seed. What is plumule? That forms future shoot. What is radicle? Which forms future root. These thick swollen structure which stores food is called as cotyledon. If these are two in number, it is called as dicot plant. If it is one in number, it is called as monocot plant. Now the axis on which the plumule and radicle are present is called as embryonal axis or embryonic axis. Now the portion of embryonic axis above the cotyledon is called as epicotyle. The portion of embryonal axis which is below below cotyledon is called as hypocotyle. So this is all about a brief description about the seed. Now we are moving to the types of seed on the basis of the food storage. So types of seed on the basis of cotyledon we have already done here. This is classification A, that if they are having two cotyledon dicot, if having one cotyledon monocot. Here we are moving with the types of seed on the basis of presence of endosperm. So if endosperm is not present in the seed and consumed during the process of development, then those seeds are called as non-endospermic seed, or it is also called as X-albuminous seeds. Like in case of pea and gram, you can imagine it in this way, like those seeds in which endosperm is completely consumed, their cotyledons are becoming thick and fleshy to store food and to provide nutrition. So all those pulses or all those plants which are having thick cotyledons will be coming under the category of X-albuminous seed or non-endospermic seed. Hope you will remember it. Now, when we talk about X-albuminous seed, here non-endospermic and X-albuminous, these are two same. One more seed we have to take here which is missing, that I'll be telling you. This is not X-albuminous, this is endospermic. Coconut, if we are talking about, this is endospermic seed or albuminous seed. It is called as endospermic because whatever coconut water you are drinking, that is actually the liquid endosperm. So this is called as endospermic seed, or it is called as albuminous seed. So coconut and maize, that corn is becoming popcorn because of the stored endosperm. So you can remember the endospermic seed in two ways. One, the coconut water. Second, the endosperm of the corn, which is making it popcorn. So this is all about the types of seed on the basis of endosperm. Once again, should I repeat? Okay, let's see. Endospermic seeds are those seeds in which endosperm persists till maturity, and non-endospermic seeds are those seeds in which endosperm does not persist till maturity, rather their cotyledons are becoming thick and fleshy to store food. Now there are some conditions given here. Briefly I am going to discuss. The condition which is required for germination is number one, water, number two, oxygen, and number three, temperature. We will be discussing the experiments based on these conditions for germination later on. A brief diagram of germination is given right in front of you, that how the germination takes place. First of all, this seed is germinating. If it is getting all these three suitable conditions, then seed coat ruptures after absorbing water, and that imbibition pressure will be generated, causing the cleavage in the seed coat. Ultimately, the first structure which is coming out is the radicle, which will form the future root. And the second structure on the top is coming out. This portion is called as plumule, which will form future shoot. And ultimately, from this small seed, a small beautiful seedling is developed, which is ready to do photosynthesis. And meanwhile, you can see the size of the cotyledon is reducing, because this cotyledon is just providing nutrition to this germinating seedling. Once the leaves are produced, there is no role of cotyledon anymore. Now these leaves are green, consisting of chlorophyll, so they can perform photosynthesis, and this plant is becoming independent and growing as a mature plant later on. Now, what is the importance of seed in the plant? As we all know, it is for increasing the generation, or you can say sustaining the life of particular species on the earth for propagation. It provides food for growing embryo, absolutely right. It protects the embryo if it is consisting of obviously the seed coat, it protects the inner delicate embryo. And seed is the mode of dispersal which will be dispersed by most of the animals, birds, and even human beings. How they are dispersing, you are well aware of this. Like if you are eating mango and throwing the seed anywhere, and it will get favorable condition and will germinate. In the same way, animals are eating lots of fruits and vegetables, and even their excreta is consisting of seed which is not digested by their intestine. So ultimately these seeds are viable enough to produce the next generation. And these agents for dispersal of seed may be air, water, explosion, or animals, sometimes human also. So here dicot and monocot, a beautiful diagram is given, which is given in your ICSE book also. Each and every part you are already familiar with, but once again I am discussing for you. This is a dicot seed and this is monocot seed. So in dicot seed, if you can see carefully, we are having two cotyledons. How will you identify? We have two cotyledons, 1 and 2. This is a unique kind of embryo of bean seed which is having folded plumule leaves. This is something unique about the bean seed. Then the plumule is the portion from which new shoot will be emerging out, and radicle is the portion from which new root will emerge out. Portion of the embryonal axis, as I discussed, below the level of cotyledon is hypocotyle, above the level of cotyledon is epicotyle. Now these two thick fleshy portions are storing food, that is cotyledon. Now there are two seed coats, testa and tegmen. So testa they have labeled the outermost, and this inner layer is called as tegmen. So testa and tegmen are actually the protective structure of seed, which are the result of outer and inner integument after fertilization. But when we talk about the maize, we can't call it as a grain. Reason behind, the maize seed, or you can say maize grain, is there because here the fruit wall and seed coat is fused together, and that is a board question. That why we are not calling it as a seed, we are calling it as a grain. So it is equally important for class 9th also. In case of maize, fruit wall and seed coat is fused together, that's why you can't separate it out, no, that's why it is called as a grain. Now here seed coat, testa and tegmen is also there. Now this complete portion which you can see, the major portion of the corn which I am making as a demarcation line, this complete portion is consisting of endosperm, and that's why it is called as endospermic seed. And this little portion which is remaining behind is consisting of rest of the parts of embryonal axis, where you can see we are having plumule for giving rise the new shoot, we are having radicle for giving rise the new root. But here this plumule is covered by another structure which is called as coleoptile, a protective covering on the plumule. You can see this is a small cover like structure which is covering the plumule. This is called as coleoptile. And when we talk about coleorhiza, rhiza means root. So root is again protected by another structure. You can draw it in this way also. So this is called as the coleorhiza, protecting root. Now where is the embryonal axis of this? If you want to draw the embryonal axis, this is the embryonal axis of the maize grain. Now, if you talk about the scutellum, that is scutellum is a rudimentary structure over here, which is called as single cotyledon. This scutellum is very important for 1 mark question. What is a scutellum? Where is it present? So scutellum is a single cotyledon which is rudimentary in case of maize, because it is already having huge amount of endosperm. So these are the labelings which you should always remember in case of monocot and dicot seed. And if differences will be asked, you can easily write that they have two cotyledons in dicot seed, one in monocot seed. Here examples are given: pea, gram, bean, all the pulses you remember, and here example is wheat and rice. Any other example, or you can say any other point can you add over here? Let's see. Scutellum is not there, like leave about scutellum, you have written here. You can go with the coleoptile and coleorhiza. Coleoptile and coleorhiza is present in case of monocot seed, which is absent in case of dicot seed. So that's all for today. We will be discussing the experiments and some more topics on the seed. Hope you will like it. If you want any other addition over here, kindly comment and inform me. Thank you.